We all start life as one single cell. Then that cell divides and we're 2 cells, then 4, then 8. Cells form tissues, tissues form organs, organs form us. These cell divisions, by which we go from a single cell to 100 trillion cells, are called growth. And growth seems like a simple thing because when we think of it, we typically think of someone getting taller, or later in life, wider. But to cells, growth isn't simple. Cell division is an intricate chemical dance that's part individual, part community-driven. And in a neighborhood of 100 trillion cells, sometimes things go wrong. Maybe an individual cell's set of instructions, or DNA, gets a typo, what we call a mutation. Most of the time, the cell senses mistakes and shuts itself down, or the system detects a troublemaker and eliminates it. But enough mutations can bypass these fail-safes, driving the cell to divide recklessly. That one rogue cell becomes 2, then 4, then 8. At every stage, the incorrect instructions are passed along to the cell's offspring. Weeks, months, or years after that one rogue cell transformed, you might see your doctor about a lump in your breast. Difficulty going to the bathroom could reveal a problem in your intestine, prostate, or bladder. Or a routine blood test might count too many white cells or elevated liver enzymes. Your doctor delivers the bad news. It's cancer. From here, your strategy will depend on where the cancer is and how far it's progressed. If the tumor is slow growing and in one place, surgery might be all you need, if anything. If the tumor is fast growing or invading nearby tissue, your doctor might recommend radiation or surgery followed by radiation. If the cancer has spread or if it's inherently everywhere like a leukemia, your doctor will most likely recommend chemotherapy or a combination of radiation and chemo. Radiation and most forms of chemo work by physically shredding the cell's DNA or disrupting the copying machinery. But neither radiation nor chemotherapeutic drugs target only cancer cells. Radiation hits whatever you point it at, and your bloodstream carries chemotherapeutics all over your body. So what happens when different cells get hit? Let's look at a healthy liver cell, a healthy hair cell, and a cancerous cell. The healthy liver cell divides only when it's stressed. The healthy hair cell divides frequently, and the cancer cell divides even more frequently and recklessly. When you take a chemotherapeutic drug, it will hit all of these cells. And remember that the drugs work typically by disrupting cell division, so every time a cell divides, it opens itself up to attack. And that means that the more frequently a cell divides, the more likely the drug is to kill it. So remember that hair cell? It divides frequently and isn't a threat. And there are other frequently dividing cells in your body like skin cells, gut cells, and blood cells. So the list of unpleasant side effects of cancer treatment parallels these tissue types: hair loss, skin rashes, nausea, vomiting, fatigue, weight loss, and pain. That makes sense because these are the cells that get hit the hardest. So in the end, it's all about growth. Cancer hijacks cells' natural division machinery and forces them to put the pedal to the metal, growing rapidly and recklessly. But using chemotherapeutic drugs, we take advantage of that aggressiveness, and we turn cancer's main strength into a weakness.
One out of every 2 men and 1 out of every 3 women will be diagnosed with cancer. But despite those huge numbers, most individuals don't know what that really means.
At the simplest level, cancer or cancer cells are cells that have lost the ability to follow the normal control that the body exerts on all cells. In our body, we have billions and billions of cells, and they have different functions. It's a very complicated process under incredibly phenomenal control. And if something goes wrong and that control is lost, and particular cells escape the normal control mechanisms, and they continue to grow, and they may spread, that's what we call cancer.
Those cells together, we would call that a tumor. Specifically, cancer is a malignant tumor, and we call it malignant because not only can it invade into adjacent organs, but unfortunately a cancer can spread to other tissues, and that can be life-threatening.
Cancer can actually occur anywhere in the body because there are cells everywhere in the body. In women, one of the most common cancers, of course, is breast cancer, and in men, prostate cancer. And in both men and women, lung cancer and colon cancer are common cancers.
It's important to understand that the cancer that occurs in one individual is very different than the cancer that occurs in another, just like those individuals are different. So a lung tumor in one person will be very different from a lung tumor in another person.
Once the diagnosis of cancer is made, of course, the next obvious question is, what do you do? There are several things that are really relevant. The stage of the cancer, which is information about where is the cancer. Do you say it's a particular kind of cancer? How much cancer is present? Has it spread? Is it in lymph nodes? Has it spread to other organs of the body?
Cancer treatment actually is very complex. And part of the reason is because cancer is this constellation of over 200 different diseases. They have common characteristics, but they're all very different from each other. In addition to that, the cancer itself is not homogeneous. There may be 3 or 4 or 5 or 6 different slight variations in the cancer cells that are there. People ask, why? Why does my cancer not go away? It shrunk by 70%. What's wrong with the other 30%? Well, it's probably a different subtype of that cancer, which is going to require a different kind of treatment.
There are 3 primary therapies for cancer: surgery, radiation, and chemotherapy. Surgery works by directly removing the tumor. The radiation therapy provides X-rays to kill individual cells, and the chemotherapy provides chemicals that can kill those individual cells, but they have side effects.
The best therapies that we can produce really are the result of optimizing the amount of tumor that we can kill by any treatment and minimizing the amount of damage that we cause to the normal cells that would be affected by that treatment.
At Cancer Treatment Centers of America, we have a very robust integrative oncology program. Integrative oncology is taking those conventional oncology treatments and integrating those with therapies like acupuncture, naturopathic medicine, chiropractic, nutrition, to blend those together and to create the most appropriate treatment plan for that individual patient at that moment in time.
Cancer Treatment Centers of America has invested in a model in which all of the effects of cancer and its therapy are aggressively treated and managed. It's not just enough to kill the cancer if we don't treat the pain, the fatigue, the depression, or the anxiety that comes with a diagnosis of cancer.
Receiving a diagnosis of cancer can be a frightening thing. The good news is that today is probably the most exciting time in history in terms of the treatment of cancer. Options that didn't exist a few months ago certainly didn't exist a few years ago, like the ability to genomically profile a tumor and to take that individualized fingerprint of that cancer may direct us to tailor treatment in very specific ways.
We believe that in the future, many more patients with a number of different tumor types will potentially be able to benefit from the advances in precision medicine.
There are very hopeful options that are available to us as clinicians to make a difference in patients' lives. And it's therefore just as important for patients to know that so that they have those hopeful options and they take advantage of them.
Okay, right here, this is our glossary of terminology we're going to be using today. We're going to add a little bit to it, so if you'll just follow along with me. First thing I want you to look at here is what the shape is of DNA. It is called a double helix. What does that word helix mean to y'all? What do you think of helical? If something's helical, what are you thinking? Huh? Helicopter. I like it. What does a helicopter propeller do? It spins. Helical means to spin. So double helix, what does double mean? Two. So double helix means a twisted ladder. And we're going to see that better next time in class. We're actually going to build a DNA model with some pieces, and you're going to be able to see that. Okay, I need everybody with me, please. Heads up, please. Thank you. Okay, so that is just simply the shape of DNA. If you will, please, write 1, 2, and 3 next to those that I have 1, 2, and 3 next to. These are the 3 parts of DNA: deoxyribose, phosphate group, and nitrogen base. And you can go ahead and write underneath nitrogen base 4 types. Okay, up here at the top, let's go ahead and write what a nucleotide is, and it will tie into those numbers we just wrote down. A nucleotide, if you'll write this here, a nucleotide is made up of number 1, number 2, and number 3. And it is the monomer of a nucleic acid. If you recall back to whenever we talked about biomolecules, we talked about monomers and polymers. A monomer, is that the single unit or the whole big thing? It's the single. Mono means one, okay? So it's the monomer. All right, so the monomer has 3 parts. Ivan, you with me? All right. So there's the 3 parts: deoxyribose, phosphate, and nitrogen bases. There are actually 4 nitrogen bases, and we'll talk about those in a minute. If you notice, deoxyribose, it says it's part of the sides, and the phosphate group is part of the sides, but then it says the nitrogen base is the rung of the ladder. What does rung mean to you? Have you ever, on a ladder, which part is? It's the part you step on. It's the steps. The rung is the steps. If you want to write steps next to rung, if you didn't know that, you can. And so when you look at the shape of DNA, you'll kind of see that they're the rung. There are 4 different bases. If you'll draw this kind of bracket and point back up to number 3 so you'll know those are the 4 types. They are adenine, thymine, guanine, and cytosine. Weird words, I know, but there you have it. Okay, on each of these it tells us that they bond with one of the others, like adenine bonds with thymine by a hydrogen bond. Thymine bonds with adenine by a hydrogen bond. Guanine bonds with cytosine by a hydrogen bond. And cytosine bonds with guanine by a hydrogen bond. So this hydrogen bond is defined right here. It is weak. It is not super strong, and that's important because DNA opens and closes a lot. So you don't want a really strong bond right there because it'll keep it from opening up, and we have to have the DNA open up. And then we have our base pair rule. Sometimes you'll see it written as Chargaff's rule. Some dude named Chargaff figured out that these things bond together. But there's an easy way to remember them, and it will be on a video here in a minute. Adenine always bonds with thymine, and you can just remember apples in the trees. Apples go in trees, apple tree. And cytosine bonds with guanine. Cars go in the garage. Car garage. CG. So that's a little memory device to help you remember who bonds together. For about the next 2 months, we're going to be talking about DNA a lot. And along with that, we're going to be talking about A's and T's, C's and G's. So it's real important that you understand who bonds with who, because that's going to come up over and over and over again. That's right. So if you'll turn your paper landscape, I don't care, if you want the spine, I've torn mine out, but if you want the spine to be the top or the outer edge to be the top, it does not matter. Just turn it that way. And then kind of over here towards like where the middle to right, we're going to write the title of this page, which is DNA. Okay, does anybody remember what DNA, or do y'all know what DNA, yes? Yes. What does DNA stand for? Nobody?
What is the first word?
D.
No, what is the first word?
Deoxy.
Never mind.
Never mind. Okay, it is deoxyribonucleic, and I'm going to focus this in better here in a minute, acid. Okay?
Ribo what? Nucleic.
I got it closer for you there.
Do you have any more glue stick? I've tried bringing my own glue stick down. There's no more.
So I'm going to enlarge out here. If you will, we're going to get some of our puzzle pieces here. Let me come out. Okay. So if you'll get one of your deoxyribose.
In the middle?
Just set it, we're not gluing it in yet. Don't glue it in. Just, yeah, some of mine aren't cut very well either. We had friends help me cut, and so, and we're going to start with cytosine. So just to make all of ours, or I'm sorry, let's start with guanine. Guanine. Okay, so these are the three parts. Remember, we just talked about that a nucleotide has three parts. It has a deoxyribose, a phosphate, and a nitrogen base. This is one of the nitrogen bases called guanine. On these puzzle pieces, I have added in some little keys to help you figure out where they fit together. Can you kind of see where they might fit together? Try it on your own. Don't glue, just move them around. See if you can fit them together.
Cytosine go with the guanine.
It will, yes.
Does it matter where we put the star on the square?
Just look, see if you can figure it out.
I can't. I did.
Did you get something like this? Now, if you notice, there is a square here, and you'll see why. But on a nucleotide, the phosphate, on this particular nucleotide, the phosphate's sticking up. The phosphate from the next one's going to fit in right there. That's why it looks that way. So this is a complete nucleotide. It has three parts, a phosphate, a deoxyribose, and a nitrogen base. In order for this to fit on our page, we're going to need to move this all the way up to the upper left-hand corner, as far up as you can go. And if you'll glue that in right now. What? I know, it's not really a puzzle, no. Well, this is how the DNA looks. I can't change that.
Get over it.
Okay.
Okay, now the cytosine goes with the guanine.
Hang tight.
She's saying bad stuff. I don't appreciate how you just called them that word. So we glue the whole thing down, right?
Yeah, glue this one nucleotide down.
She has glue stick.
All right. Some of you have already noticed, due to the puzzle piece-like shape, that cytosine does match with guanine, and we learned cars in the garage. So the cytosine is going to match there. Something weird happens on the other side, though, of the DNA. Everything is flipped. So get your cytosine and kind of match it up to the guanine. Get you another deoxyribose and a phosphate. And this side is going to be flipped upside down like this, so that this phosphate is pointed down. So once you have that kind of set up, you can glue that portion down. And mine aren't cut very well, which yours may not be, but that's okay.
Whoever cut these gave poor effort.
Well, it was your classmates that cut it, so be forgiving of them.
All of these? I highly doubt you would have put more effort. We cut the blue crosses. Yeah, so whoever cut these, the purple ones did good. I'm sorry.
All right, this is what we call a base pair. We have two bases that are paired together, so this whole section together is called a base pair. Do y'all see, look over by the door, that turquoise looking chart that says human genome landmarks? That is actually the human DNA mapped out. And some of those strands of DNA, like number 1 has something like 265 million base pairs on that one strand. So that's 265 million of this on one strand of DNA. And that is just one strand. That's like the strand from mom. Then you also have that same strand from dad. And all of that fits inside the nucleus of every one of your cells. So you can see the real thing can't be this big. This is frankly a model, right? But I just wanted to get you a visualization of how many base pairs there are in your DNA. Has anybody figured out how the next one would fit in? Okay, so your phosphate would go on the square, and if you overlap them, that's good. And then the star, and then you can choose. I'm choosing adenine so that the print will be upright. Adenine right there. You can glue that down. Da, 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 da. And then if you've figured out the pattern, what matches with adenine? Apples in the trees. Thymine matches together. Again, this side is flipped upside down.
My star is missing.
Oh, did it get cut off? Oh, stick it up underneath the deoxyribose. There you go. There we go. So this is, you should have something that looks like this.
Unfortunately, my square didn't make it to the next square. Somebody, yeah. Buttercup. She needs to go learn how to cut. No, just kidding.
You do it yourself next time when she says the question.
What? She said you can do it yourself. I would. Why that's not a job. Exactly. Well, do. That moment when you're trying to be kidding, but you think it's serious. Yeah. And it just turns awkward in like 4 seconds.
She said she'd kill us. I got you.
If for some reason your shifting kind of throws it off the edge of the page, that's okay. Just take your little phosphate and bend it under so it doesn't stick out. It'll be just fine. That's just great. All right. So let's label a few things here. Grab your pen or pencil. This bond that holds these two guys together right here is called a hydrogen bond. That's on your glossary. I'm going to label both of them. This one I'll just abbreviate H bond. Hydrogen bond. This guy right here is one of the 4 nitrogen bases. Yep. Nitrogen base. Okay. The nucleotide, remember, is made up of 3 parts. Look back at your glossary. What are the 3 parts that it's made up of? It is made up of Deoxyribose sugar, a phosphate group, and a nitrogen base. So here they are. Don't write until I've completed this. We're going to circle a nucleotide. So it's going to include that, and these, and that. So now circle those three things. Okay, so this is a nucleotide. And it has three parts. We're just going to list them. It has the phosphate. It has a deoxyribose. And it has a nitrogen base. What do we call this? The single unit of DNA. It is a monomer. This is the monomer of DNA, a nucleotide. And then just to remind us of those nitrogen-based pair rules, let's write this. Cytosine. Am I on the screen? Yeah. Yay. Okay. Cytosine to guanine and adenine to thymine. Huh? Adenine. It's written over here in the orange too, if you can't see my spelling. All right, so the nucleotide has three parts, and there's a really great way, a little memory device I like to use to help us see the nucleotide. So if you'll turn to a blank page, you can either turn to the next blank page, or if you don't, or on the back of this, I don't care. And hold your hand out like this. Just hold it up in front of you. I'm just holding it on the screen. Okay. The nucleotide has three parts. If I told you that the phosphate is represented by my thumb, what do you think the palm of my hand is representing? The deoxyribose. So what would my fingers be representing? The nitrogen bases sticking out over here, right? And how many nitrogen bases are there? 4. And how many fingers do you have? 5. No, 4. You have 4 fingers to remind you that there are 4 nitrogen bases. So just kind of as a memory device, we're going to draw our hand. If you want to trace your hand on the page, you can.
So I can do
Or,
it this way instead
or if you, whichever way, or if you just want to draw a hand, I'm going to trace because I'm a very bad artist.
I have to draw my left hand because I right.
And don't put your fingers together so it'll make it a little easier to see. Don't spread your fingers apart.
No turkey.
No, we're not making a turkey.
That's so 4th grade.
I'm going to kind of draw my fingers in here.
I remember used to draw it and put beans in it.
So there's my hand. And just so you don't open up your notes and say, why did I draw my hand on my notes? Let's title the page,
That looks like your paper saying, boy.
My Hand is a nucleus. I know, doesn't it look bigger when you draw it? Like, I think my hand looks skinny, and then I draw that, it looks like fatness. Okay. So my hand is a nucleotide. So what did I tell you your thumb is? Phosphate. It's a phosphate.
You create a wolf out of this, a dog.
Okay. And what's the palm of my hand? The deoxyribose. And then my fingers is the nitrogen base. And how many nitrogen bases are there? 4. So we just remind ourselves with 4 fingers. So that's a little memory device. Throughout the unit, I might go, okay, what are the parts of the nucleotide? And you're going to hold your hand up, and it's actually a cheat sheet that I'm going to allow you to use on the test. You can be sitting there and test and go, oh, my thumb's the phosphate, and my palm is deoxyribose, and my fingers are the nitrogen bases. Yay, me.
Today's topic is the cell cycle and mitosis. But before we get into the cell cycle and mitosis, we want to make sure and talk about why these words are important and how they relate to real life. So if I say the word cancer, this word probably means something to you. Either you know somebody who's been diagnosed with cancer or you've heard about it on the news. There's many different types of cancers, but one thing they all have in common is that they are caused by uncontrolled cell growth. That means somebody's own cells grow out of control. Now, that doesn't mean that there aren't other contributing factors. For example, for some cancers, having genetic links, meaning it runs in the family, can make somebody more likely to develop it. Or having excessive exposure to UV light for some cancers. Those are risk factors that can increase your risk, but the major cause of cancer is uncontrolled cell growth. That also means it's something that is not contagious, and it's something that anyone can develop. You have to realize that all the cells in your body are very, very busy, and they're either in one of two different phases. They are either in a phase called interphase, and during interphase, they are just growing, they're replicating their DNA, they're doing their cell functions, or they are in mitosis. Mitosis is a fancy word that means that they are dividing, they are making new cells. Well, if you think about the cell cycle, whether they're in interphase or mitosis, it's kind of like a pie graph. They're spending about 90% of their time in interphase. Most of the time, cells are not dividing. Most of the time, cells are in interphase. They're growing, they're doing their daily cell functions, and they're replicating their DNA. Most of the time, cells are not dividing. Now, depending on what kind of cell, it might do mitosis more or less often. For example, your hair follicle cells, they do mitosis frequently, which is why your hair can grow up to half an inch every month. But other cells, like, for example, your brain cells, your neurons, some of them actually do not perform mitosis, which is why if you have some type of major injury to the spinal cord or to the brain, sometimes those do not heal very well because those cells, they do not replicate. So mitosis is very important in order to generate new cells, which can help with growth. And also, if you damage your cells, if you're walking down the street and you have some kind of accident and you skin your knee, well, you're going to need to repair those damaged cells. You're going to need to be able to do mitosis to make those new cells. The thing is, you don't want your cells to always be doing mitosis, because if they're always doing mitosis, then they're always dividing, and then they're not carrying out their actual function. And that's a problem. That's where checkpoints come in handy. See, along the cell cycle, there are checkpoints to check that the cell is growing correctly and replicating its DNA correctly and doing everything it's supposed to do correctly before it divides. And what happens if the cell fails its checkpoint? The cell does something called apoptosis, which basically means the cell destroys itself. It self-destructs. This might seem kind of harsh, but actually it's really important because it ensures that you don't have these cells that are messed up continue on and divide. You don't want damaged cells to continue on and divide because they might not do what they're supposed to. They might start to grow out of control. They might not be regulated correctly. That's where cancer cells come in. See, cancer cells, as we had said, are your body's own cells that are growing out of control. So they're not following the rules of these checkpoints. They're flying past the checkpoints, going through the cell cycle, and frequently doing mitosis, which means that they're dividing very, very often. And so by doing that, they can grow into these things called tumors, which are really a mass of cancer cells. So we're not really sure what causes them to escape these checkpoints. There's a lot of risk factors that a person can have that can increase their risk of this happening. But when these cells get past these checkpoints and grow out of control, they become a problem because many times they can travel through the bloodstream. They start growing out of control. The body can't identify them and destroy them with the immune system. So this becomes an issue. And usually one of the treatments that somebody who has cancer will undergo is chemotherapy. And this works by targeting cells that go through the cell cycle quickly and hit mitosis quickly. And that's one reason why somebody often will lose their hair when they're on chemotherapy, because they're on this medication that's targeting fast-growing cells, and your hair follicle cells are also fast-growing cells. Cancer cells have some other factors that make them difficult to treat. One is that cancers have the ability to secrete their own growth hormone. This is very bad because growth hormone, in a sense, think of it as a hormone that says, feed me. And what happens is the blood vessels will divert over to those cancer cells and supply the cancer cells with nutrients. This is not good because this means the cancer cells will have the nutrients they need to keep growing out of control. It also can take nutrients away from nearby healthy cells. There's some medications that are being researched that maybe could stop the growth hormone from cancer cells. And in a sense, that would be great because the cancer cells would not be able to get the nutrients to grow out of control anymore. Of course, you have to make sure that the medication is targeting just the cancer cells and that you're not affecting the growth hormone of nearby healthy cells. You wouldn't want that. So it's kind of a tricky situation. So let's recap what we talked about today. We talked about the cell cycle, how cells are more frequently in interphase than they are mitosis. Interphase is when cells are replicating their DNA or they're growing, carrying out cell processes. In mitosis, the cells are actually dividing. We talked about how during this cell cycle, there's checkpoints that regulate the cell and keep the cell from being able to continue on if it's damaged. Unless we're talking about cancer cells, which are cells that have escaped the checkpoints and are growing out of control. Be sure to check out our clip on mitosis. This process is really fascinating, but it's also a really important one in order to understand how your body cells work. That's it for the Amoeba Sisters, and we remind you to stay curious.
Hi, it's Mr. Anderson. Today I want to talk about DNA. Before we get into transcription, translation, mitosis, meiosis, Mendelian genetics, all of these things are based on DNA, and so we should have a real good understanding of what DNA is. Now, this animation here is just gorgeous as DNA spins, and it gets this reputation for being a kind of a mysterious, magical kind of a molecule. But it's actually pretty simple compared to proteins. It's pretty straightforward, and so you should understand the parts of it. And so basically, the building blocks of DNA are nucleotides. And so just like carbohydrates are made of sugars and protein is made of amino acids, the building blocks of nucleic acids, both DNA and RNA, are going to be nucleotides. And so here's one nucleotide right here. A simpler one, if we were to look at basically the building blocks of RNA, you've seen something that looks like a nucleotide before. Because if you take the adenine and a ribose sugar and then add 3 phosphates to it, then you have ATP. So you've seen something that looks a lot like this. But a nucleotide has 3 parts to it. So part 1 is going to be this purpley part. And that's going to be what's called a nitrogenous base. And so in DNA, there are 4 types of nitrogenous bases. This one pictured here is called guanine. But there are 3 other types in DNA. So you've got guanine, you also have cytosine, thymine, and adenine. So when you see these letters in DNA, what they're really referring to is this nitrogenous base. And the reason it's called a nitrogenous base, just look at all the nitrogen that we have inside there. So that's a nitrogenous base. They come in 2 different types. Some of the nitrogenous bases, like this one, are called purines. Purines are going to have 2 of these carbon chains. The purines are going to be guanine, so that'd be one purine, and then the other one's going to be adenine. We'll also have some that are a little simpler. They're not quite as big, and those are going to be called pyrimidines. Pyrimidines. The ones in DNA are going to be cytosine and thymine. And I'll show you those in just a second. So the first part of a nucleotide is going to be the nitrogenous base. The second part is going to be this sugar right here. And in DNA, that's called deoxyribose sugar. Now in RNA, that's going to be a ribose sugar, but in DNA, it's going to be a deoxyribose sugar. And the reason why is if you were to look right here in a ribose sugar found in RNA, there'd be a hydroxyl group coming off of it. But in DNA, you're missing that oxygen, and so we call it or missing oxyribose, so deoxyribose sugar. So that'd be the sugar right here. And then the third thing that's going to be found is a phosphate group. And so a phosphate group, we're familiar with that from ATP. That's going to be a phosphate group right here. And so those are the building blocks of nucleotides. And so let me get my writing out of the way, and we'll go to the next slide. And so now this is maybe a little bit more familiar when you're looking at the structure of DNA. And so this right here, this nucleotide that we just kind of went over the parts of it, that nucleotide is going to be found right here. Now, one more thing I should mention about nucleotides is that if you look up here on DNA, there's a 3' end to DNA, and then there's a 5' end. And so what does that mean? Well, that's going to refer to the sugar itself. And so the sugar itself is going to have, since carbon's so ubiquitous, or it's found everywhere in organic material, we don't even draw the symbol on here. So there would be a carbon right here, a carbon right here, a carbon here, a carbon here, and then a carbon here. And so we simply number those carbons. And so this carbon right here is called the 1' carbon. This one is called the 2' carbon. This one right here would be the 3' carbon. This would be the 4' carbon. And then this would be the 5' carbon right here. And so the 3' carbon is going to be coming off this side, 5' coming off the other side. And so basically, when you're looking at DNA, this right here would be the 3' carbon, and this down here would be the 5' carbon. So when you hear DNA and the idea that DNA flows from 3 to 5', well, if you look at all these deoxyribose sugar with the oxygen on the top, they're going to flow in this direction from 3' to 5'. But if you look on the other side of the DNA, it's going to go in the opposite direction. So it's going to go in the 3' to 5'. So DNA is said to be anti-parallel. So it's going to flow in one direction on one side, and it's going to flow in the opposite direction. They're parallel to each other, but they're flowing in opposite directions. And so if we were to look across from the 3' end here, we're going to have a 5' end over here. Now, why is that important? Well, when you're building DNA, when we're going to add another nucleotide on, so here's one nucleotide, here's another nucleotide. When we're going to add another nucleotide, we can only add it on the 3' end. And so I could add a new nucleotide on this side, but I can't add it to the 5' end. And so when we get to DNA replication, how DNA copies itself, that's going to be really, really important. We could add one down here to this 3' end, because this is going to be our other nucleotide right here. So we could add one here, but we can't add it to the 5' end. Okay, so enough with the 3' and the 5'. Let's talk about some other the large parts of DNA. If we look at the backbone, so the backbone is going to be on this side and on this side, and it kind of, DNA looks like a ladder. In other words, if this is the backbone, then the rungs of the ladder are going to be the nitrogenous bases that go right down the middle. But if we look at the backbone itself, it's simply a deoxyribose sugar, a phosphate, a deoxyribose phosphate, deoxyribose phosphate, deoxyribose. And so the back is going to be the same with every nucleotide. And if we look on the other side is going to be phosphate, deoxyribose phosphate. It's going to be the same thing on the other side. So the backbone is going to be relatively boring. That's not too exciting. But if we look to the inside, that's where we're going to have these purines and the pyrimidines, these nitrogenous bases. And if you look right here, the adenine, which again is a purine, so it's got these two rings, that's going to be connected using these hydrogen bonds to the pyrimidine on the other side. And if we have a purine on this side, we're going to have a pyrimidine on the other side. And there's base pairing. In other words, adenine is always going to bond to thymine. And if we look down here, guanine is going to bond to cytosine. Or this would be a cytosine and a guanine, and this is going to be an adenine and a thymine. Now, what do I mean by they bond to each other? Well, the bonds are going to be right here in the middle. So if you look at DNA, structure of DNA, every one of these atoms is going to be connected to every other atom by a covalent bond, except if we look right down here in the middle. If we look right down in the middle, these are actually hydrogen bonds. Remember, hydrogen bonds are very weak. So these are relatively weak bonds that go right down the middle. Why is that important? Well, if I pull DNA in either direction like this, the DNA will unzip in the middle, and we're just breaking those hydrogen bonds in the middle. If we let go of it, it'll just go right back together again because those hydrogen bonds are going to form. And just like adenine is covalently bonded to the nucleotide below it, it's hydrogen bond between the two. And so that'll be super important, like when we're doing DNA replication or when we're making messenger RNA, these hydrogen bonds, super easy to break. And so that would be the gross anatomy of deoxyribonucleic acid. Those are going to be the parts. What's important is that we can store information in here. So when we talk about transcription and translation, every 3 letters here are going to code for 1 amino acid. And so if we were to break down the word deoxyribonucleic acid, you should be able to understand where that name comes from. And so in other words, the deoxyribo part comes from this. It comes from that deoxyribose sugar. The nucleic part comes from the idea that it's found inside a cell. The DNA is going to be found inside the nucleus of the cell. And then the acid part actually comes from the phosphate group. Phosphates are going to donate hydrogen, and so that's going to make it acidic, which will become important in just a sec. So here's our DNA. It's just repeated nucleotides over and over and over. But we know that DNA looks like this. And so DNA is going to have this 3-dimensional shape. And scientists think that RNA was the first genetic material on our planet, and then DNA is kind of an upgrade to that. And so if we see this is our DNA, it's essentially that same ladder, but that ladder has been twisted into a helix. And the reason why it's not really drawn here is that there are also going to be hydrogen bonds that are holding each of these. So there's going to be hydrogen bonds here and hydrogen bonds here. And so basically what that does is gives it this 3-dimensional shape, and that makes it really, really stable. And so DNA has some advantages over RNA. Number one, it has a more stable 3-dimensional shape. But the other nice part is that we could have a mutation on one side of the DNA, and since mutations are passed from generation to generation to generation, if it's in DNA, then we have a backup copy on the other side. So if there's a mistake here, we can actually look at the DNA that's on the other side, and then enzymes can actually cut this out and replace it on the other side. And so that's DNA. Those are the major parts of DNA, and it's really not that complex. If you understand breaking it down to a nucleotide and the idea that it's simply just a ladder of information, and that information essentially tells a cell how do you make a protein. And so that's DNA, and I hope that's helpful.